Hello, everyone. My name is Christopher Anderson, and I work with the Environmental Engineering Biotech Laboratory at West Virginia University under Principal Investigator Dr. Emily Garner. Today, I'm going to be talking about the bacterial environment in drinking water distribution systems. So first, we should probably define what a drinking water distribution system is. Drinking water distribution includes the process of water going from a source, such as a ground aquifer, a lake, a stream, a river, into a treatment plant, then through a system of distribution networks and plumbing, onward into a building or premise, and then finally delivered through a tap to a consumer. So you might ask, well, where does bacteria come into play? Bacteria exists all around us, and in fact, it's not all bad. Some bacteria are actually beneficial to natural processes in the environment. Specifically, drinking water distribution systems are well suited for low nutrient requiring bacteria called oligotrophic bacteria. Despite treatment and disinfection, oligotrophic bacteria that are typical of nutrient depleted drinking water can actually grow and colonizing within plumbing as water travels from the treatment plant to the consumer. These bacteria can colonize in one of two ways. Either they can exist in the bulk water of the drinking water distribution system, or they can collect in the form of biofilms. Biofilms are a collection of bacteria that form a small community that surrounds itself with an extracellular polymeric substance that kind of acts as a slimy adhesive to keep those bacteria together. For example, if you've ever seen pink or orange buildup near your drains in your shower, or on your shower head for that matter, you're actually experiencing a biofilm of collected bacteria. It should again be noted here that this isn't necessarily harmful to your health. In fact, most of the bacteria in your drinking water is harmless. However, biofilms and bulk water both can become harmful in the event that they are infiltrated by an opportunistic pathogen, which is a disease-spreading bacteria, such as Legionella pneumophila and Mycobacterium avium. A more recent example of this is the 2014 Flint, Michigan water crisis, in which a switch of the source water caused an outbreak of a pneumonia-like illness known as Legionnaire's disease, which is caused by Legionella pneumophila existing in the drinking water. This example, along with others, reiterates the importance of clean, safe drinking water as it pertains to public health. Hence, understanding the composition and diversity of drinking water distribution system bacteria can then hopefully give insight to factors that might influence the microbiome, whether they're from physical flow of water, chemical reaction, or biological factors that are interacting within biofilms and bulk water alike. And so there are a few primary objectives with this research. First, we wanted to determine the overall bacterial taxa, or what exactly exists within these distribution systems, and then also calculate diversity within the samples to see, from sample to sample, how different the bacterial environment is, or if it's predictable based on, certain physicochemical factors. In addition to that, we look to find potential correlations between the presence of opportunistic pathogen bacteria such as Legionella pneumophila and Mycobacterium avium compared to the rest of these samples' composition and abundance levels of other bacteria. That way, we could learn if the presence of one bacteria is indicative of a lesser presence of another bacteria, perhaps an opportunistic pathogen. Overall, this research can be used to increase the awareness of the importance of drinking water microbiology to prevent crises such as what occurred in Flint, Michigan, and by that also obtain a greater understanding of factors that may or may not influence pathogenicity in drinking water distribution systems. In order to complete these studies, genetic sequencing was completed in a previous sampling by my principal investigator, Dr. Emily Garner, that targeted the 16S rRNA gene within the drinking water samples. From there, a bioinformatics software known as Quantitative Insights into Microbial Ecology 2 was used to project and analyze the genetic sequences from the provided data set, and then cluster those sequences into groups with 99% similarity, which is what we call an operational taxonomic unit. In other words, if two sequences of DNA were 99% similar, they got grouped together. And then those were compared to the green genes database to assign taxonomy or naming to those bacteria so they could be properly identified. Then alpha and beta diversity were calculated within the QIIME2 software, and the compiled data following that was also imported into R to look at non-metric multidimensional scaling, to see what potential significant drivers of the bacterial community might have an effect of the overall composition and diversity within them. A Spearman rank correlation was then used to see if there were any correlations between bacteria and the pathogenic strains of bacteria, or any notable significant correlations if they could be made. And so to simplify a bit, we basically just took raw genetic data from these drinking water samples and then took that data into a high performance computing system to then do quality and trimming, which was then used to cluster these sequences together and assign taxonomy, and then furthermore do these analyses to see really what's going on within the drinking water distribution system. So to summarize our results a little bit, you can see in figure 1, which is a taxonomic bar plot that shows the relative abundancies on a 100% scale for each of the samples in utility A and B. Now, it should be noted for this figure that utilities C and D were omitted just for simplification, and so it could be more easily readable, which in this case, it's definitely hard to see, and I would encourage whoever's listening to go to the poster and zoom in and look for yourself. But of the 4 utilities that were sampled, the 4 drinking water systems that were sampled, only utilities A and B, as you can see in figure 2, were sampled for biofilms. Now, in this taxonomic bar plot, the most notable observed phylum was Proteobacteria for both utilities A and B. This is not necessarily of concern, but it could be, considering that Legionella pneumophila is actually part of the phylum known as Proteobacteria. Figure 2 shows a box and whisker plot that shows the diversity of samples at a local scale using the Shannon Alpha Diversity Index. And basically what that means is a higher index could mean both that there is a greater number of species present, which is called richness, and also that the species are more evenly proportioned, which is known as evenness. Again, you may want to look at the actual poster and zoom in here to see this a little bit better. While figures 1 and 2 can be used to get more of a general idea of what's present in the drinking water distribution system as far as bacteria are concerned, figure 3, which is a non-metric multidimensional scaling plot overlain by canonical correspondence analysis, is used to show beta diversity between the samples. Unlike alpha diversity, beta diversity shows more of a holistic representation of the data set. This is because it looks at the relative similarity between each individual sample. And so a shorter distance between 2 points on this graph indicates a greater similarity in the overall bacterial community structure of the 2 samples. As it is currently shown, the plot separates these samples based on utility, but this can be done in a multitude of different ways. This was just the most convenient to show. What's interesting about this graph is that it's overlain by a CCA plot. And so you'll see these arrows on this plot all point in a specific direction of the graph. And they can be used to identify potential drivers of the bacteria community. In other words, what actually causes those samples to have those bacteria within them. It should be noted here that in order to be more stringent and to have a more clear representation, these environmental variables were restricted at a significance level of p less than 0.01. Notably, we can see that the type of disinfectant residual, in other words, what disinfectant is left within the distribution system, could be a notable or a key potential driver of microbial community structure. This makes sense as free chlorine and monochloramine residuals are typically opposing treatment methods. In other words, you don't ever have a free chlorine residual and a monochloramine residual at the same time. It's usually one or the other. As you can see, both of these arrows are also longer than most other arrows in the chart. This means that they are more significant than shorter arrows. And specifically, the monochloramine arrow points more in the direction of those sample communities represented by utility A, whereas utilities B and C specifically more align with the free chlorine residual. And so this at least goes to suggest that the bacterial community differs with treatment methodology. This graph can be viewed in a multitude of different ways, and so we are continuing to look at all the other variables or variability within the graph itself. Lastly, with figures 4a and 4b, we looked at completing a Spearman rank correlation to look at the correlations between at the genera level with Legionella and Mycobacterium, respectively. At this point in the research, we have actually become a little bit stumped in terms of these graphs or these representations, these correlation graphs, because what we were hoping to see was something specific stand out, whether it be a correlation with a specific genera that could be associated with the presence of Legionella or Mycobacterium, or whether it's a negative or positive correlation. However, for both instances, the correlation coefficients with the significant variables, or excuse me, the significant genera were all relatively weak between 0.2 and 0.4. And also to this end, none of these significant correlations were found to be negative, which is actually what we would probably hope for in this study. Because ideally, in a drinking water system, if you could find a genera or a specific type of bacteria that would be almost a preventative measure against an opportunistic pathogen genera such as Legionella, then you could interpret that in a way where you might want to stimulate the growth of that specific genera that could help prevent a pathogen like Legionella pneumophila in the case of switching a water source, maybe like what happened in Flint, Michigan. And so with that, we are going to need to do further analyses in order to see what relationships are occurring here, and if there are any relationships that can in fact be manipulated in that way. Now that we're at this point, we're looking at a lot of different options for what we can maybe do next in order to make some sense of where we're at with this data. So to conclude a bit, the purpose of this study overall is just to guide subsequent experimentations. And what I mean by that is that there's a lot of data here. And I'm sure to someone who's new to this type of, or to this area of work, hearing me spew words about all of these different correlations and everything may not have as much significance, which in reality is similar to the way we feel about the data. The overarching goal is to find some hint or some guidance within this multitude of data so that we can manipulate a specific variable in a laboratory in an experiment that might prove, for example, that the occurrence of this bacteria hinders the occurrence of Legionella pneumophila. Or similarly, a physical condition of the water, such as the flow rate or a chemical residual, and how it might affect the presence and abundance or the ability of opportunistic pathogens like Legionella and Mycobacterium to thrive in the drinking water environment. So to that end, the majority of this research so far has been searching for clues to then allow us to make more specific conjectures and more direct experimentation later on in the lab. With that, future iterations might include more of an emphasis on biofilm sampling, as this was potentially a caveat to the dataset that we used for these analyses. As you could see, utilities C and D did not have biofilm samples, and so maybe getting a more overall conjecture between biofilm versus bulk water might be a route that we could take in the laboratory later on. And then we can see how those differ from each other. Overall, there's increasing relevance with this type of research, especially as we move forward in the world and freshwater resources become more and more limited. And so there's an innate need in the scientific literature to continue research like this so that we might preserve all of those freshwater utilities that we have. As freshwater becomes more and more limited in the world with increased population density and whatnot, it is therefore extremely important to safeguard the water quality of our existing drinking water distribution systems. Overall, we hope to continue to seek understanding within this specific data set and then later on guide subsequent experimentation. At the same time, we hope to increase the awareness of this issue when it comes to water quality as it relates to human health and public health specifically. And that's all I have for today. So thank you for tuning into this presentation. And if you do have any questions, please ask. I know that there is a lot of information here and a lot of small questions that could probably be answered to help anybody's understanding of this presentation. Thank you.